Hey everyone, Mr. Jennings here. Today on Big Orange Biology, we're going to go over the FRQ number 1 practice problems I assigned this week. This week's FRQ came from 2017's exam administration. So this one here is publicly available. It is released on the College Board website. It takes some digging to find, but you can find it. However, before you do the questions, and I know the answers are out there and the answer, I do recommend and I encourage you to actually take the test question seriously and do it on your own before you actually watch the rest of this video. So I know several of you all have already done that. Not everybody has. Okay, so today's question is going to be about caffeine and bees. So it's actually a behavior problem, but you really don't need to know a lot about behavior to answer these questions. So this is one I feel like, outside of one major component of the question, would work fairly well for the FRQ revisions that are happening this year on the exam, where there's only 2 FRQ questions, one being of a longer length and the other being of a shorter length. So when you take this question, you should take about 25 minutes to complete it. So keep that in mind. You need to get this done in about 25 minutes or less. So that said, let's go over the question. Okay, so here's the deal. The question here, I actually have a new interface trying this out. This is Microsoft Whiteboard for those who are actually interested in this. But this is a new interface, so we're going to see how this works. And what we have here are several different questions that deal with, the main prompt. So this here is what the AP exam used to look like. It's obviously changed a little bit even before this year's, all the dramatic changes. So we're going to read through the question and make sure we understand the prompt, understand what the questions are asking, and we're going to go over the graph. And I want to try to emphasize which ones you're going to see on the AP exam. I think I can actually probably hide myself for this part. So let's start over here. It's working on the Zoom feature. Okay, so questions 1 and 2 are on free response. Okay, this here is just gibberish. This was not even on y'all's practice test. Okay, so table 1. In flowering plants, pollination is a process that leads to fertilization of an egg and the production of seeds. Some flowers attract pollinators, such as bees, using visual and chemical cues. When a bee visits the flower, in addition to transferring the pollen, the bee can take the nectar from the flower and use it to make honey for the colony. Nectar contains sugar, but certain plants produce caffeine in the nectar. Caffeine is a bitter-tasting compound that can be toxic in high concentrations. To investigate the role of caffeine in nectar, a group of researchers studied the effect of 0.1 millimolar caffeine on bee behavior. The result of the experiment is to test the effect of caffeine on bees' memory of a nectar source as shown in Table 1. All right, now let's talk about the actual questions. Question 1. So this is the one question I feel like will not be assessed the way it is on the AP exam, because the question seems to be favoring the concept more or less, not so much about your ability to make a graph, but ability to read a graph. But I think the ability to read a graph and make a graph go hand in hand, and be able to understand how to interpret a graph really means you need to be able to construct it. So that's what we do. We're still going to practice it. And also, a lot of the practice problems for these type of FRQs are less graphic interpretation and more graphic building. So we got to make sure we have that. So on the axis provided, you have to construct, and I'm going to highlight this part here. All right, so when you're asked to construct, that means you're going to have to build a graph. And the big thing is you have to have a appropriately labeled graph to illustrate the effects of caffeine on the probability of bees visiting the nectar source. This is known as memory. So they are going to give you, if they are to do this, you will likely not get a grid. They can still ask you to make a graph. It's highly unlikely because of the digital interface. I don't know how this is going to look. However, they could, in theory, have you post a picture, take a picture with your phone, upload it. Highly unlikely. But when we talk about constructing a graph, you're going to usually get a grid provided to you. And the big parts are you need to know that all graphing questions like this are worth 3 points. So usually you have to construct a graph. I'm freehanding this on a digital tablet. So graphs usually can require a few components. Let's just make this a little bit bigger. You need to have you have to have a bar graph in this situation. Now they're going to go, why is this a bar graph? It's not a continuous range of data. It's two pockets. It's categorical data. So because it's categorical data, you're going to use a bar graph. So your y-axis is going to be the time. So this here is 10 minutes, 24 hours, and this here is appropriately termed memory. So if we were to call this anything, this would be your short, and this would be your long. Over here on the y-axis is going to be the average probability of revisiting nectar source. It's a really lovely and lengthy title. So having your axis labeled and appropriate bar graph is going to get you 2 points. Okay, I just paused to double check myself here. Yeah, a correctly plotted bar graph will get you 1 point, and appropriate labels, units, and scaling will get you another. Typically, when you have appropriate units, you also usually have to title the graph. And the graph here would be just the title of the question here: effects of caffeine, probability of bee revisiting. Okay, so you can write that as the title. And this is basically it. Now, the next thing is you have to have a bar graph that's appropriately scaled. As we go from 0 to 1, that's our scale. So our two groups here, we're going to have two different bars. We're going to have a control bar. So we're going to make two different lines. We're going to do a green line for our control. Our control is at 0.72. So on our bar graph, 5, 0.7, 0.8, 0.9, 4, 0.2. So we're going from 0 to 1. Our first data point is 0.72 for control. Control is in green, so we're going to go to, and we're going to continue on the other control for the 24 hours. So there's our two controls. And then we're going to go back and do the caffeine. Now the caffeine for 10 minutes was at pretty good high probability of returning. And 24 hours it had a very slight drop of 0.7. So that is the bar graph. Now that is 1 point as well. So we have appropriately drawn bar graph. We have a appropriately labeled units and scaling. The third point comes from the error bars. So this plus or minus system right here, that's our error bar. So for error bars, I'm going to use black just so it pops a little different here. So control was 0.09. That's very little overlap. And the caffeine at the 24 hours, 0.07. So it's again very tight. And the other one's, all right. Now, one way you could go about this is if you wanted to, you could probably draw this. I've already made the graph, so I had to went with it. Now, let's talk about B. So this here was all 3 points. So let's talk about part B. Part B asks, based on the results, describe the effect of caffeine on each of the following. So there's two different events. You had to describe the effects on short-term memory of the nectar source and long-term. So I'm going to write this to you in kind of a grid-based. You all are to write this in more paragraph, but you had to have something along the lines of these two phrases. So for the short term, you should have seen that the bar graph, that caffeine had little to no effect. Caffeine had little to no effect on short-term memory. That's the observation you should have noticed, and I'll point out the graph here in a second. As for long-term memory, you have, there's obviously has caffeine greatly improves long-term memory. So let's talk about where we got those results. All right, so caffeine over here, the numbers, the bar graphs are pretty close. The error bars, given them out possibility of, graphing process, these are too close to call. Meanwhile, the 24 hours, there is a stark 9-day difference between the two. The error bars are nowhere near each other. The data is almost twice as much. It is clear and abundant that caffeine has a positive influence on the long-term memory of the bee's nectar source. So that's where the answers came from. Now, part C. See the one that, yep. All right, I'm going to highlight some parts here for C. C has a problem that I noticed some students had issues with because it's clear in the text, but it's easy to overlook the phrasing. Okay, so C asks, design an experiment using artificial flowers to investigate the potential negative effects of increasing caffeine concentrations in nectar on the number of floral visits by bees. So you have to design an experiment. Awesome. That's not hard. The key part, though, is a lot of kids were wanting to write how the experiment affects bees, and a lot of them were talking about like the mortality of bees, because in the FRQ prompt, it mentions that the caffeine is toxic to bees in high doses, but we're not talking about high doses. So we're talking about increasing the caffeine concentration, but they're looking at not the number of bees alive, but look at the number of bee visits. Probability. Higher probability means higher visits. So you had to identify. Now when it comes to identification, they're asking you to identify 3 things: null hypothesis, control, and the predicted results. This type of question, by the way, and also this one, All three of these are more likely to be on the FRQ that you receive on the exam. Question 1 part A, less likely. You've already provided the graph, and you'll be doing more with B, C, and D. So let's talk about C. You had to identify the experimental value. So C. You had to identify the experiment. Now the experiment called for 3 components: the null hypothesis, the control, and the results to refute. Again, I'm presenting this to you in a grid. You are to write this in a more paragraph prose style format. Do not write in bullet point. Do not write in grid. The grid allows me to just give you highlights, the things you look for in your own writing so you can score yourself. Things to look for and the things they're looking for. It is not, and I repeat, it is not the appropriate way to answer them. So in this experiment, you were asked to develop a null hypothesis about the effects of increasing caffeine on the effects of increasing caffeine on bee visits. The null hypothesis, if you cannot remember, and just don't remember, is the hypothesis without any, basically the idea that the independent variable does not affect. In this case, the independent variable was the increase of caffeine. The dependent variable is the number of bee visits. So you would write this, null hypothesis. The idea would be increasing caffeine concentration or amounts does not impact number of visits. So increasing the caffeine does not affect the impact of bee visits. Does not impact the number of bee visits. Instead of impact, you can write effect. But that's the answer they're looking for. That's the null hypothesis. Basically, that any relation between this data is merely circumstantial. It is merely coincidence. It's merely just randomness. That's the null. Now, the reality is usually you have a null and you have an alternate hypothesis. The alternate hypothesis is what you usually are used to proposing as a hypothesis. Now, let's talk about the control. In this experiment, we're looking at the amount of caffeine concentration. So an experiment would be no caffeine, because we're looking at the effects of caffeine on bee visits. The best control you can have is just have no caffeine and see what normal bee visits look like. Then you increase the caffeine in the nectar and you just track it. Now, when it comes to designing the experiment portion, you could write out a little bit more about this, but the 3 things you have to have are these components. So you could write something along the lines of you put the nectar in various flower-like receptacles and you count the number of bees, and each receptacle has a different concentration, all to answer the question and to address the null hypothesis, and you identify a control. The result that you would see to refute the null, well, there's 2 results you could look for. The first result is number of bee visits increase with caffeine concentration. That's expected. That would be the next thing that would definitely refute the null hypothesis. Increasing the caffeine does impact the number of bee visits. The other alternative is there's a difference to, basically, you got results that varied from the control. The control is no caffeine in the nectar. So if I remove the caffeine, I'm still getting the same visits as increasing the caffeine. That means caffeine wasn't there. But if I saw increasing bee visits with the caffeine versus no caffeine, obviously I reject the null. All right. Now that sets us up for part D. All right, let's look at part D. So researchers found that nectar with caffeine tends to have a lower sugar concentration than nectar produced with caffeine. So by making caffeine, you have less sugar than those without. Plants use less energy to produce caffeine and nectar than they do to produce sugar. So we're going to propose 1 benefit and 1 cost. So at this point, I meant to tell you this, B, these were 1 point each. And C, besides that being an OR situation, these are 3 points. So currently we're at 3 points for A, 2 points for B, 3 points for C. So D, you have to propose 1 cost and 1 benefit. The benefit for the plant and the cost for the bee. So we're going to kind of do this in a little bit of so we're going to look at the cost to the bee and benefit. So cost to the bee. First off, there's less sugar in the caffeine. So the cost would be less sugar to the bee. That's the obvious one. What that actually translates into, and the correct answer would be this thing here. Increase energy expenditure. Increase energy use. Less quality honey. Because that's one of the things they do with these nectar. They actually take a little bit of the nectar for themselves to get to the flight. But in reality, what that nectar is going to be done is it's going to be regurgitated back in the hive to help produce honey. That's quality honey. More trips, so that's one of the reasons you have increased energy use. They're going to have to take more trips to get the same amount of sugar. it's just overall less efficient for the. But the benefit to the plant is massive. This energy used, meaning more energy to carry out function. And now they have all this extra energy to carry out their functions. They're able to carry out and work out, so the plants can carry out more functions, live better, and they are all getting more pollinators because, hey, increased caffeination creates more of a drive to visits to the plant. It's kind of like you're purposely making the bees slightly addicted to the caffeine, so they're kind of constantly visiting the caffeine source, so they have more memory of it. All right, let's kind of summarize here. So, on the AP exam this year, you're only going to get 2 FRQs. One of those FRQs is probably going to look similar to this question that you received. However, it's not 100% the same. You're likely not to be making a graph. No, it's possible. It's highly unlikely. The data currently emphasizes they interpret data, interpret a graph. So that's more relevant. However, parts B, C, and D are clearly stated as something that you would be asked to do on the AP exam. You're going to be asked to identify parts of an experiment, design experiments. You're going to be asked to make predictions, to identify and describe possible trends in the data. And also, you need to identify costs and benefits and any kind of changes and disruptions. So all these here are available and likely to be on the. That's it, though. Other than that, I hope you're all having a great time.